that is here in this time zone with us. Thank you for assisting to the Engineering and Sciences Congress of the UABC, CICITEC. I am pleased to present Dr. Tin Wui Wong with his key conference, Non-Destructive Biomedical and Pharmaceutical Research. He's from the Non-Destructive Biomedical and Pharmaceutical Research Center, Smart Manufacturing Research Institute. And the title of his key conference is Oral Inhalation and Skin Drug Delivery Innovations for Cancer and Diabetes Treatment. I will present a brief profile from Dr. Wong Tin Wui. He obtained his PhD degree from the National University of Singapore in 1999. He's presently the lecturer and principal fellow at the Non-Destructive Biomedical and Pharmaceutical Research Center, Smart Manufacturing Research Institute, Universiti, Teknologi, Mara. His research areas are primarily focused on particle scaffold design for oral, transdermal, and pulmonary drug delivery, and design of pharmaceutical processors for innovative dosage from manufacture. He has published over 120 peer-reviewed articles, and he is the editorial board member of Asian Journal Pharmaceutical Science Associate, editor of Drug Development and Industrial Pharmacy, Drug Design, Development and Therapy and Technology in Cancer Research and Treatment, and regional editor of Current Drug Delivery. Professor Wong is the founder of Non-Destructive Biomedical and Pharmaceutical Research Center, Malaysia, and co-founder of Sino Malaysia Molecular Oncology and Traditional Chinese Medicine Delivery Joint Research Center, Medical College in Jiangsu University, China. He serves as a visiting professor of UCSI University, Malaysia, and National University of Singapore, also lecture professor of Jiangsu University, China, and adjunct professor of Nirma University, India. I leave the microphone to Professor Tin Wui Wong. Thank you and welcome.
Thank you, Dr. Gonzalez. First of all, I wish to double check if you can hear me clearly and the screen is seen.
Yes, we can hear you and we can see the screen.
All right, so thank you. So good afternoon, everyone. I'm Tin Wui from Malaysia. First and foremost, I would like to thank Prof. Louis and the organizing committee for the kind opportunity for me to be here now to share with you some of our latest research findings on oral inhalation and skin drug delivery innovation for cancer and diabetes treatment. So this afternoon, I'm going to first highlight about the skin drug delivery, where in our laboratory, we focus a lot of work on cutaneous melanoma, which is a form of skin cancer. Now, this cutaneous melanoma first is embedded in the epidermis. Now, as times go by, when the disease gets more severe, you can see that the melanoma can actually penetrate deep down into the dermis and subsequent metastasize into the systemic organs. Now, there are many drugs that have been used in treating cutaneous melanoma. Now, 5-fluorouracil is a conventional drug with a very good economical value. Now, it has been found in the latest clinical trials whereby it can also be equally effective in treating cutaneous melanoma. However, skin drug delivery is met with a few challenges pertaining to the treatments of cutaneous melanoma. First, there will be a skin barriers for drug delivery. Second, when come to the melanoma, the cellular membranes act as a barriers, hindering the drug delivery into the intracellular compartment. Now, if you know the skin very well, basically it is constituted of 3 layers: epidermis, dermis, and subcutaneous tissue. Now, the topmost layers of the epidermis, which is known as the stratum corneums, is the primary drug penetrations barriers. Now, commonly, the drugs penetrated into the skin via 3 routes. First, it may be diffused through the epidermis and stratum corneum. Second, the drugs may diffuse through the hair follicles as well as the sweat glands. Now, as what we know, the hair follicle and sweat gland only occupy 0.1% of the total skin area. Hence, majority of the transport pathway is still relying on diffusions across the epidermis and stratum corneum. Now, knowing that stratum corneum is the main hurdles in drug penetrations through skin, now let's take a look on its anatomical structures. Now, stratum corneum basically is constructed in a very systematic manner, which is known as a bricks and mortar. Now, it is constituted of keratin-rich corneocyte embedded in the lipid matrix. Now, if you look closely on the pink color lipid matrix, you will see that it is actually a lipid bilayer alternating with the aqueous pores. Now, the lipid bilayers consist of cholesterol, fatty acid, and ceramide. Now, do note that although the lipid bilayer is fatty and lipidic in nature, it does contain polar moiety such as OH, NH, as well as carbonyl bond, C double bond O. So the transport of drugs through the stratum corneums can either be mediated through transcellular or intercellular routes. Now, from this anatomical structure, you can imagine how challenging it will be for a drugs to penetrate through the skin. Now, under transcellular routes, the drug needs to go through layers after layers of corneocyte. The drugs needs to move into the lipid zone, followed by corneocyte, and moving out from one corneocyte to another corneocyte. So this is very, very challenging. Now, likewise for intercellular routes, the drug needs to go through the aqueous pore or lipid bilayers in the intercellular pathway. Now you can imagine that the diffusion pathway is rather tortuous with twist and turn. So both trans and intercellular pathways are not exactly easy for drug transport. So to overcome these challenges over the past 20 years, passive and active technology have been invented. Now, passive technology basically refers to dosage form or drug delivery system design. So that may involve hydrations of the skin, whereby the water can engorge or enter into the aqueous pore, making the aqueous pore bigger, allow more drugs to go through the skin. Alternatively, the dosage form may be formulated with chemical penetration enhancer such as oleic acid, ethanol, and surfactant. Now, this enhancer can actually fluidize the lipid or even extract and remove the lipid from the intercellular pathway, making the aqueous pore large enough for drugs penetration. Of course, nanocarrier being the latest favorite of all scientists, it can be made of waters, oleic acid, ethanol, and surfactants, in addition to matrix materials such as polymer. So the nanocarrier have the ability to fluidize the lipid or even remove the lipid from the intercellular pathway. And nanocarrier by itself, as the name suggested, it has a nano geometry. It is small enough to penetrate through the intercellular route. And active technology has been invented to be used on the skin prior to the applications of passive medicines. So the traditional method includes stratum corneum removal by the adhesive tape. Of course, the latest technologies, they are microneedles, electrical methods such as iontophoresis and electroporation, ultrasound such as sonophoresis, photomechanical wave, and pressurized gas. Now, with the advance of nanomedicines, you may thought of a delivery of drugs through the skin can make it easy. However, this is not entirely true. Now, a systematic review by our laboratories indicated that nanomedicines may help skin drug transport, but it is rather complicated and affected by the size and zeta potentials of the nanoparticles. Now, if you look at the relationship between the particle size as well as skin drug transport's propensities, you realize that a lot of nanoparticles, they are small, but they do have a very poor skin drug transport, in fact, right? Likewise for zeta potential. Now, it is known that the positively charged or negatively charged nanoparticle can actually promote skin drug delivery. Now, positively charged nanoparticles, it tends to attract to the anionic lipids of the skin. hence facilitate their penetration into the skin. Now, for negatively charged nanoparticles, it tends to repel with the anionic lipids of the skin and making the skin fluidized and softer, and the aqueous pore become larger, and this will facilitate the drug molecules that are released from the anionic nanoparticles into the deeper layers of the skin. However, the relationship of zeta potentials with the skin drug transport is not universal. You can see that vesicular carriers such as liposome, transposome, polymosome, and niosome, ettosomes, they do not have a specific dependence on zeta potential in skin drug delivery. So to overcome all the heavy dependence of size and zeta potentials on skin drug delivery, now the active technology is invented, which I've described earlier, like microneedle, electrical method, and ultrasound method. Now in our laboratory, we actually innovate microwave as the active skin penetration enhancer. Now the ideas come about because in the past 20 years, we have been using microwave to develop solid dosage form such as tablet, bits, pellets, granules, and microparticles. Now we realized that treating solid dosage form by microwave, they will change the conformational flexibility of polysaccharide, protein, and lipid materials. Now, knowing that the skin is made from lipids and proteins, so we will wonder if we treat the skin with microwave, will they soften the skin? Will they porosify the skin? Will they fluidize the lipid and make the aqueous pore larger? So with this query in mind, we then move on to conduct a preliminary study. We actually irradiate the surfaces of the skin with microwave. Now you can see that the untreated skin is actually wrinkled in structure. That could be related to the bricks and mortar systematic structures of the skin, of the stratum corneum. Now treating the surfaces of the skin with 2450 megahertz for the right time, 5 minutes, you can see that the entire surfaces of the skin turn smoother. So we presume that the microwave can actually destroy the stratum corneum structure temporarily, right? This will allow the medicines, the nanomedicines to go through the skin. Yeah. So having these positive preliminary trials, we move on to create a small projects. Now in this project, we intend to deliver 5 fluorouracils, which is a conventional therapeutic that has been found to be useful in cutaneous melanoma treatment. Now 5-fluorouracil, although it's a very small molecule, but due to its polar natures, it's very hard to go through the skin. So delivering 5-fluorouracils through the skin is a great challenge. And in this study, we intend to deliver the 5-fluorouracil into the skin and being retained in the skin instead of going into the systemic circulations, which will elicit adverse effect. And we formulate the 5-fluorouracils into the form of attosome. So there are 3 variants of attosomes are being formulated, E5, E10, and E15, with 5 to 15% of ethanols. Now you can see that through the drug release and drug permeation studies, we realized that the E5, the attosome with the lowest content of ethanols, it appears to be the best dosage form to carry the 5-fluorouracils into the skin. Now, the E5 induces a lower rise in systemic level of the drugs, and it induces the highest level of skin drug retention. Now, this is not surprising because the E5 attosome with 5% ethanol, so it actually slightly larger in size, and they are more negatively charged. So although they can fluidize the skin, but due to the larger nano size and more negatively charged, it tends to be accumulated in the epidermis, hence having a higher drug retention in the skin. Unlike E10 and E15, perhaps E10 and E15, with the higher ethanol content, they actually fluidize the skin extensively, and the attosome actually transport across the epidermis into the dermis and disappear into the bloodstream, right? Which is unfavorable with regards to systemic adverse effect. Now, with this positive positive attributes of the E5, you then may ask, well, why not we prepare E1 or E2 with just 1 to 2% of ethanol? And this perhaps can promote a further skin penetration of nanomedicines. Now the answer is not, because ethosomes with the lower content of ethanols, it may be less elastic. Hence, it lacks the flexibility to squeeze through the intercellular roots into the deeper layer of the skin. So this gives us a good reason to combine ethosome with microwave technology. So we first treat the skin with microwave, followed by the applications of the ethosomes E5. Now you can see that with the right frequency and the right timing of treatment, there is a negligible rise of the systemic blood levels. However, the skin drug retentions is being promoted. Right? So having this very positive combination usage of ethosome and microwave, we begin to investigate what are the reasons behind all these observations. Now to do that, we actually evaluate the skin microstructures upon the application of ethosome, treating with microwave or combinations of both. Now we examine the lipid regime and protein regimes of the skin, as well as the epidermis and dermis zones of the skin by means of thermal analysis and infrared analysis. Now, allow me to ignore all this number, and I shall present to you a schematic diagrams to illustrate what is the general findings. Now, in the earlier slide, I've highlighted that the skin is characterized by stratum corneum, which is a very robust drug penetration barriers. Now, it's keratin-rich corneocyte, now separated by intercellular pathway, which is lipid-rich with aqueous pores. Now, all this stratum corneum and viable epidermis is supported by the dermis below of the skin. Now, when we treat the skin with microwave, a few things happen. Now, first, the microwave will interact with the keratin of the corneocyte. Now, the keratins will actually transform from alpha helix to beta sheet. Now, as the results, the condensation of keratin occur. Now, once the keratin condense, you can see the corneocyte, the size turns smaller. So the corneocytes will equally shrink and condense together with the keratin. So the microwave can also interact with the intercellular lipids, fluidize them, and soften them. As a result, the summative effect brings about an enlargement of the intercellular pathway. Now, with this intercellular pathway being enlarged, the ethosome, the drug molecule, can then penetrate through the skin easily. And microwave is actually an irradiation. Now, it actually irradiates volumetrically. Now, apart from interact with the epidermis, with the stratum corneum, now microwave can also penetrate deep into the dermis and interact with the extracellular protein. Now, from our study, it is tentatively concluded perhaps the extracellular elastin has been swelled by the microwave and forming a dermal barrier, which prevent the ethosome and drug molecule from transporting further into the bloodstream. So with this combination effect, you can see that the ethosome and drugs can penetrate through the skin, but it cannot enter into the deeper layers of the dermis and systemic circulation. Hence, the skin drug retention is promoted, but the systemic permeation is not enhanced. So this is entirely the pictures and the favorable scenario that we want in order to treat skin melanoma topically. Now, we further prove our in vitro findings by the in vivo studies. Now, you could see that the rats with the skin treated by microwave, followed by the ethosome E5, they have a very little drugs appearance in the plasma. So a lot of drugs are being retained in the skin, which is ideal for topical treatments of cutaneous melanoma. Now, in the next slide, you will see the actions of ethosome and microwave on the melanoma itself. Now, ethosome and microwave by themselves, they can actually kill the melanoma cell line, and combination of microwave with ethosome, it actually promotes the cytotoxic actions synergistically. Now we examine the melanoma cell lines by means of thermal analysis and infrared analysis. Now you can see that the attosome, microwave, and especially the combination use of microwave and attosome, they can actually fluidize the lipidic layers of the cancer membrane. So this will allow the attosomes to enter into the cancer cell and exert the cytotoxic action. Now, through confocal electron microscopy, you can see that the melanoma cell line treating with microwave followed by the attosome has a lot of attosome being entrapped in the cancer cell. So this in turns enhance the cytotoxic action of the 5-fluorouracil. Now, through the use of a series of metabolic inhibitors, we also realized that the cellular uptake of attosome under the influence of microwave is mediated by the lipid raft pathway, right? The endocytosis pathway. Now, we further the experiments by developing solid nanoparticles apart from the earliest liquid attosome that we have described. Now, we realized that chitosan 5-fluorouracil conjugate solid nanoparticles perform similarly to the liquid system. Now, you can see that treating the skin with microwave for a specific duration, they can actually enhance the penetrations of the nanoparticles into the skin. And one interesting features that we have found out is that the chitosan nanoparticles actually fluidize the palmitic acids zone of the stratum corneums, whereas the microwave actually fluidize the ceramide 2 regions of the stratum corneum. So it's exactly because chitosan nanoparticle and microwave fluidize the different regimes of the lipids materials of the stratum corneums. Hence, this explains why when we combine the usage of nanoparticles with the microwave, it brings about a synergistic cytotoxic action. Now, with reference to chitosan nanoparticles, the similar finding is found whereby the combination use of microwave with nanoparticles can promote the skin drug retentions, which is good for topical treatments of cutaneous melanoma. And likewise, in the melanoma cell lines, the combination of microwave with the chitosan-based nanoparticles actually promote the endocytosis by lipid raft pathway. So in one of the latest work that we have conducted pertaining to melanoma, we begins to look into the systemic effect of the cutaneous melanoma. So we expect that the melanoma will metastasize. So in order to have the nanomedicines to be able to detect the metastasized cancer in the systemic organs, we actually conjugate the oligochitosans with the folate, which is the targeting ligands. Now, throughout these studies, we realized that a folate content about 20% to the chitosan-based nanoparticles is essential in order to bring about a maximal cellular uptake by the melanoma and cytotoxic actions through S-phase cell cycle arrest. Now, so much so about cancer. Now, in our lab, we're also very active in studies of diabetes. So there's a project whereby we intend to deliver macromolecular therapeutic insulins through the skin. Now, conventionally, the skin formulation of insulin is developed through the use of fatty acids such as oleic, linoleic, and linolenic acid. However, now in our study, our main conclusion is that the fatty acid actually does not promote transdermal insulin delivery. In fact, it demotes and reduces transdermal insulin delivery. But microwave, on the other hand, is different. Now, you can first treat the skin with microwave, followed by the application of insulin. Now, we found out the microwave actually promote transdermal insulin delivery to a very significant extent. So this indeed is a good news for the diabetics because injection is really challenging and induce a lot of psychology threats to the patients. Now, why microwave perform well compares to fatty acids in terms of skin drug delivery of insulins? Now, we actually characterize the skin in many ways, and one of the parameters that we have conducted is transepidermal water loss. Now, we realized that treating the skin with fatty acid or microwave, they can actually promote the skin permeability. So, however, only skin treated by microwave has improved and increased insulin penetration. Now, why is that so? Now, the reason is very simple. Now, according to our investigations, now fatty acids such as oleic acid, they may be able to interact with the lipid bilayers of the stratum corneum, fluidize them. Now, theoretically, this should allow insulin to penetrate through the skin, but practically not. That is because oleic acid, although it is a penetration enhancer, but its depositions in the intercellular roots will create a hydrophobic barrier. And these hydrophobic barriers will deter and hinder the penetrations of insulin, which is a polar, large molecular weight therapeutics. So on this note, we summarize that the microwave is much better as a skin penetration enhancer for insulin than those of fatty materials. Now let's move on to inhalational drug delivery. Now we have recently started the inhalational studies, and the study is rather simple. Basically, we have chitosan as the polymer backbone, and then from there we conjugate the chitosan with the drugs of interest, and as well as targeting ligands such as folate and other functional excipient. And we later transform the nanomaterial into nanoparticles with the targeting ligand decorated on the surfaces. And these nanoparticles theoretically should be inhaled by pulmonary routes for the treatments of lower lung cancer. However, nanoparticles being a very small particles, although it can be inhaled easily, but it will be exhaled equally fast. So 80% of the doses may be exhaled. So on this note, we actually design microcarrier to carry the nanoparticles into the peripheral and deep lungs. Now let's take a look on the way we create the microcarrier technology. Now over the past 10 years, the nanoparticle has been converted into the microparticles, which is known as the nanoagglomerate, nanocomposites, nanoparticles loaded in sugary microparticles. But because of the processing involved certain forces and certain sticky materials, so this nanoparticle is very hard to be redispersed and release the drugs to the intended cancer site, right? And furthermore, due to the strong processing strategies, the nanoscale may be distorted, and it will affect the propensity of nanoparticle interacting with the cancer cell and the subsequent biological action. So with this shortfall and limitation of the past research, we then try to hypothesize which way will be better in order for us to deliver the nanoparticles into the deep and peripheral lung. So we have chosen to physically blend the nanoparticles with a larger particle that can act as the carrier of the nanoparticles. Now, first and foremost, we actually hypothesize perhaps it's good to blend with a coarse particles, which have few hundred micron size. But think about it, it is not ideal because the coarse microparticles will only be retained at the upper part of the gastrointestinal tract. Now, should the nanoparticle fail to detach from the coarse microparticles, there will be no nanomedicine delivered to the lower lung. Hence, we actually designed the microscale particle, which is just a few micron in size, and we mix both nanoparticles with the microscale particles. Now, you can see in the next slide, both nanoparticle and microscale particle, they are very cohesive and aggregative. Now, blending of both in a specific weight ratio will have the nanoparticle sit on the surfaces of the microscale particle and mutually reduce the cohesiveness. So the entire products become very free-flowing with a low Cow index and Hosner ratio. And aerosolization and inhalation of this physical blend will then have the particle which are small enough to enter into the lower lung. And in the lower lung, the nanoparticle can detach from the surfaces of the microscale particle easily because it is just a physical blend, it's not encapsulation. And this will then serve the purpose to deliver the drugs to the lower lung for the treatments of cancer. Now, to prove that, we actually prepare solid chitosan nanoparticles by spray drying techniques. We have 6 variants of chitosan nanoparticles with a different chitosan molecular weight, chitosan concentration, or different excipients such as surfactants and ethanol. Now, we characterize all the chitosan solid nanoparticles, the size, zeta, polydispersity index, circularity, roughness, and crystallinity. Now, these parameters are characterized because they are the parameters that will have a strong influence on inhalation. You can see that the nanoparticles are generally spherical. However, the microscale particle, which is developed from lactose and PEG hybrid, they are not spherical. They are rocks shaped, which give a good aerodynamics properties to carry the nanoparticles into the lung. Now, in this development of microscale particle, we use a PEG because PEG is a waxy polymers combination with lactose can actually reduce the hygroscopic of lactose through increasing or maintaining the crystallinity of the particles. And do note that this microscale particle is only a few micron size, so it has a very good properties for inhalation. A blending of microscale particle and nanoparticle in a specific rate ratio, and we conduct the lung inhalation studies by cascade impactors. So we realized that the nanoparticle zeta potential, the size, as well as the circularity play a very important role to dictate its inhalation efficiency. Now, if you look at these schematic diagrams, generally it will tell you that the nanoparticles, they are large and round. They have a lower specific surface area. So this nanoparticle tends to be less aggregative between the nanocarriers. And likewise, nanoparticle with a high zeta potentials, they tend to repel between each others, and they will have a less nano aggregation. So most of the nanoparticle will actually deposit on the surfaces of the microscale particles. This will allow them to inhale together with the microscale particles into the deep and peripheral lung. On the contrary, nanoparticles, they are small, they are less circular, they have a larger specific surface area. So these nanoparticles tends to aggregate among themselves instead of depositing on the surfaces of the microscale particles. And nanoparticles with the lower zeta potentials, they cannot, or they have a less capacity to repel between each other. So this nanoparticle tends to aggregate together instead of depositing on the surfaces of microscale particles. As a result, their inhalation's efficiency is lower compared to the larger nanoparticle with the higher zeta potential. Now, in another experiments, we also look into inhalation of liquid medicines such as nano emulsion. We actually developed 3 generations of nano emulsion. Now, first generation is very simple. It's just the oil and water emulsions with the therapeutic dispersed in the oil globule oleic acid. Now, the second generation, we begins to advance the design. Now, we decorate the oil globules with chitosan. And the third generation, we developed the oil globule with chitosan folate conjugate. Now, these chitosan and folates are used because in this particular study, we actually would like to eradicate tubercle bacilli that is hidden in the macrophages of the alveoli, right? Now, in order to do so, we need to target the macrophage. So chitosan itself, as well as folate, it has been advocated as the good targeting agent for the alveolar macrophages. Hence, it will enable us to bring the drugs into the macrophage and kill the tubercle bacilli that induce tuberculosis. And if you look at the first to third generation, obviously the third generation works better because it has 2 targeting agents, dual receptor targeting, that can detect the macrophage to a greater extent, right? You can see that the third generation's nano emulsions, it can enter into the macrophage to a greater extent due to the dual receptor targeting. And the overall, the skin drug retention is higher with third generation's nano emulsions, which is expected to lead to a better treatment effects in tuberculosis. Now, coming to our last sections on oral drug delivery. Now, our laboratory has been working on oral drug delivery for the past 20 years. And colon cancer is one of our specific interests in terms of research. Now, in this colorectal cancer topic, we invent oral colon-specific drug delivery system. Now, this is one of the earlier designs that we have, whereby we invent in vivo intracapsular coating technology instead of fluid-based or immersion coating technology. So we coat the drugs particles at the in vivo level instead of at the in vitro or benchtop levels. Now, how to achieve that? Now, basically, in this study, we use 5-fluorouracil as the drug of interest, and we encapsulate the drugs in the pectin-rich pellets, and we fill the capsules with the pectin-rich pellets together with the coating material, which is made from ethyl cellulose and pectin. Now, upon ingestions of the entire capsule orally, now in the gastric regions, the dissolution medium or the biological fluid will enter into the capsule and wet the pectin. Once the pectin turns soluble, it becomes adhesive and sticky. Now, it will stick the hydrophobic ethyl cellulose together with the pellets and form a very robust and hydrophobic plaque. Now, this plaque is very robust and resists to disintegrate and dissolve the drugs into the gastric and small intestine. So as a result, we can then prevent premature drug release. And majority of the drug is released in the colonic regions. This is because in the colon, we have a lot of microbial enzymes released from the microorganisms. And this microbial enzyme can actually digest the pectin in the pellets as well as pectin in the coat materials and disintegrating the entire structure, releasing the drugs in the colon for treatment of colon cancer. So you can see that this is a very simple design, but yet it works even at the in vivo levels. Now, the intracapsular coated pellets, apart from being able to kill the colon cancers, to reduce the size, to reduce the number, and to reduce the histopathological grades of the cancer, what I wish to highlight to you is that one interesting feature about the intracapsular coated pellets, it can actually also reduce the average number intensities of aberrant crypt foci of the colon, which is the precancerous lesions of the colon, compared to the unprocessed 5-fluorouracil as well as the uncoated pellets, right? Now, this is very important. It shows that the intracapsular coated pellets can actually eliminate cancer as well as precancerous lesions that will develop into the cancer in one day. And in another studies, we actually challenge ourselves. Now, in this study, we developed the uncoated pellets in order to deliver the drugs to the colon. Now, how to achieve it? The judicious choice of the excipient is very important. We must have the right excipients in order to form the pellets and the particles to carry the drugs. So in this study, we use the alginate as the matrix materials. So alginate is the polycarboxylic acid. So it has a lot of COOH functional moieties. And we incorporate the alginates, the matrix with ethyl cellulose and calcium phosphate. Now note that the ethyl cellulose is hydrophobic. It can then prevent early drug release in the gastric regions or even in the earlier intestinal region. Now for the calcium phosphate, it is a moderately water-soluble calcium salt. So upon transit from gastric to intestinal regions, this calcium phosphate will gradually dissolve and release to cross-link the alginate matrix into a denser microstructure. So with this denser alginate microstructure, they can actually first capture the ethyl cellulose in the pellets, hence preventing the loss of ethyl cellulose. Second, so with the denser alginate microstructure, it can also prevent further drug release to the external medium. So it is only when the pellets transit from gastric into intestine, followed by colonic regions, then the entire structure will break down with time and release the drugs in the colon. Now, in our laboratory, so apart from the oral cancers, the colorectal cancer, we also look into oral insulin delivery, which is a hot topic and is very challenging to be executed. So as challenging as the transdermal insulin. So in these oral insulin delivery projects, we adopt alginate as a matrix material for one simple reason. Now, if you look at the ethnopharmacology, now you realize that the alginate is actually a sugar absorber. So consumption of alginate is expected to absorb the sugar, absorb the glucose from the intestine, hence can prevent absorptions and prevents the increase in the plasma sugar, right? So on this note, we adopt alginates and we intend to complex the alginate with the insulin to form the nanoparticles. Unfortunately, alginate insulin complex is very robust. They will not release the insulin at all. So on this note, we actually synthesize alginate carbon 18 conjugate and we complex the insulin with alginate carbon 18 conjugate into nanoparticles. Now, this complex nanoparticles is better in the sense that it can reduce the release of insulin in the early part of gastrointestinal tract, and at the same time, it can actually release when needed, commonly upon the enzyme digestions of the alginate, right? Now, in order to further protect the alginate insulin nanoparticle from the early digestion by gastric acid, we actually embed the nanoparticles in the calcium alginate bits. Now, note that this bit is made from calcium alginate. So with the aims to introduce more alginate that can also at the same time absorb the sugar in the gastrointestinal tract. Now we further protect the bits with a coat, which is a tripolyphosphate cross-linked chitosan oleate conjugate. Now ingestions of this bit system or coated bit systems now is met with a low insulin release in the gastric and with more insulin being released in the small intestinal regions. Now overall, it brings about a good blood glucose lowering and increase the oral insulin bioavailability. But what I wish to highlight to you, if you look at the drug release study in vitro, only about 20 to 30% of the drug is being released. So through using zeta-sizer analysis, we realized that in the intestinal regions, the coat will be eroded, the calcium alginate will be dissolved. They actually release the nanoparticles in the intestinal regions, apart from 20 to 30% of the insulin. Now, these nanoparticles, they are able to cross the mucosa of the intestine and enter into the intestinal cell prior reaching the systemic targets. Now, so it is because of the nanoparticles design itself, alginate carbon 18s complex with insulins and transform into the nanoparticles that are able to bring across the insulin through the intestinal barriers into the systemic circulation. Now, in the same diabetic project, we also look into the small molecular drugs. Now, in these studies, we actually isolate the vitaxins from the Ficus deltoideus, which is natural herbal plants. And we actually mix the vitaxin with alginate and stearic acid and spray dries into nanoparticles. Now, knowing that nanoparticle itself has a large specific surface areas, now it tends to release the drugs in the gastric. And in this study, we wish that the vitaxins can be delivered into the small intestine in order to interact with the alpha-glucosidase to prevent carbohydrate breakdowns and increase in glucose level in the blood. So on this note, we actually compact the nanoparticle with polyethylene glycol into a tablet, into a mini tablet. And this mini tablet is filled in capsule and ingested orally into the in vivo animal system. Now you can see that the, the entire dosage form design is rather effective. We have a minimal release of vitaxin in the gastric region. Most of the drug is released in the intestinal regions. And you may wonder why. Because nanoparticle upon compaction, wouldn't it the nanoparticle being fused into a lump? And hence it's very difficult for the drugs to be redispersed and released. So the tricks is in the entire exercise, stearic acids remain the most important excipients in order to maintain the dispersibility of the nanoparticles in the intestine from the compact as well as drug release. Now let's take a look at the mechanistic diagram. Now you will see that in the gastric regions, due to the PEG and the alginate carbon 18, the drug release is rather minimal, but upon prolonged transit into the intestinal regions, the polyethylene glycol will dissolve and release the PEG and release the nanoparticles. The nanoparticle may be in the clumpy form, but due to the stearic acid that can exert particle dispersion or particle wicking property, it can actually disperse the particle and perhaps even destroy the particle structure, releasing the insulins releasing the vitaxin into the intestinal regions to interact with the alpha-glucosidase. So this is the entire mapping of the mechanisms of the nanoparticles. So with that comes to the ends. Again, I wish to thank Prof. Luis for the kind arrangement and opportunity, as well as the organizing committee, my postgraduates, students, supporting staff, supplier, and manufacturers. So with that, I end my presentations. Thank you.
Thank you, Dr. Wong. It was a very nice presentation, very eloquent and complex. And this is the time for questions and answers. Is there any question, comment, or remark from the public? Please, you can write on the chat or raise your hand to open your microphone. Es momento de preguntas. Si hay alguna duda o comentario, pueden escribirlas en el chat o pueden levantar la mano para abrir su micrófono. Pueden hacer su participación en español o la pueden hacer en inglés. Si es en español, nosotros intentaremos lo mejor posible traducirlas para el Dr. Wong. Entonces, cedo la palabra al público. So with this message, we have some comments from Daniel Zarate. Very interesting presentation. We thank you, Professor Wong. That's the comment from Daniel Zarate.
Okay. Thank you.
Mm-hmm.
So actually, they can write to me if they have any questions.
Mm-hmm.
And now it's having COVID, but Malaysia, we foresee that the COVID may be able to control by end of this year. So hopefully in the future times to come, we can have more activity between the two countries. So feel free to write to me if you have any question pertaining to the presentations, or you have interest to do attachment or study in Malaysia.
Okay, thanks a lot. We will take your word into consideration. Dr. Luis Villarreal is raising hand. Dr. Luis, please.
Thank you, Tin. I just want to thank you your participation in this Congress. We're very, very grateful. And I want to let you know that we are very grateful because you have been helping us a lot for the international participation, and you give us a lot of recommendations to invite people to join to this project that we are growing in the Receipt Journal and the CICITEC Congress. We are growing, and we want to thank you publicly that you are one of the most active members of the team. So in personal, that your work is interesting with me because I also work with drug delivery systems, but I use electrospoon nanofibers, and also we use that technique of electrospraying. So have you used the electrospraying technique?
I have recently developed a small project using that. And definitely, I think Prof. Luis, I think after this conference, perhaps we can have an online discussion. Malaysia, we're actually very keen to work with your team on the pharmaceutical and biomedicals platform.
That's great. Thank you, Dr. Tin, and we will contact you later.
Okay. Thank you.
Okay, thanks, Dr. Luis Villarreal. There's another question from Jose Manuel Cornejo Bravo. Do melanoma cells behave as normal skin regarding permeation?
Okay. So in this study, you may realize that we actually look into the skin barrier as well as the cancer cell membrane barriers. So because when you apply a medicines onto the skin cancer, apart from the detectable skin cancer, the surrounding area of the skin cancer underlying in the dermis, they may have some cancer. So we may apply the medicines on the skin cancer itself, which is the dark spot. At the same time, we also may apply onto the surrounding skin areas of the melanoma. Let me just, I hope I answer your question. So we may apply the drug on the skin cancer as well as the surrounding area, because the surrounding area underneath, they may have a melanoma that has been grown into the dermis.
Perfect.
Yeah.
Okay, is there any other question, comment, or remark regarding Dr. Wong presentation?
Generally, the audience may be surprised when we use microwave. Now, for a simple, when we use microwave, because microwave is known to be a kitchen appliances, they may actually cook the skin. So just wish to highlight to you the power that we use is only 1 milliwatt. It is not 900 watt. So 1 milliwatt. Yeah.
Okay, perfect. There's another question, a follow-up question from Jose Manuel Cornejo Bravo. Do you have a model for melanoma?
Do I have
a model for melanoma? That's the question.
Models. Actually, for study, we use the SKMEL cell line from ATCC. Yeah, and for the animals models, we have yet to develop. We have developed before, not through genetically engineered mice. So what we have done is we prepare a melanoma cell line suspension, and prior to injecting under the skin, we actually give immunosuppressant drugs to the rats, to the healthy spread dolly rats, followed by injecting the melanoma cell suspension. Now, the cancer developed for a very short time, but subsequently, it appears that the rats is strong enough to kill the cancer. Yeah. So you can see that from this study, although we failed to develop the in vivo melanoma models, but you will realize that the healthy rat actually is very hard for us to induce cancer. So in future, if you wish to conduct animal study, perhaps you need to purchase the genetically engineered system or animals. They'll be easier for you to develop cancer.
Okay, thank you, doctor. There's a hand raised from, I think, Jose Manuel Conejo Bravo. Please, if you like, open your microphone.
Yes, thank you. Very interesting presentation, and I am the one doing these questions because I'm testing in system for release drugs into melanoma.
Okay.
We just started. We have a student that just started a project. I work with Dr. Villarreal.
Okay. Yeah,
So
actually, melanoma is
that's why I'm so curious. Interested in the topic. So we may talk to you later afterwards. Thank you.
Thank you.
Okay, thank you, Professor Cornejo. Is there any other question, other comment or remark from the public? Please let us know in the chat or raise your hand to open your microphone. We have a space for one final question or comment. And then we will start the next round of thematic forums. Okay, Dr. Luis Villarreal is writing on the chat, inviting to the next thematic rooms, material sciences. There's also mechanical design and manufacture and nanotechnology. Whatever you're interested to, you can enter. Hay otras, está poniendo el doctor Villarreal, unas ligas para entrar a las mesas temáticas. Pueden darle clic en la mesa que a ustedes les interese. Empieza a las 4, es decir, 1 minuto. Okay, Dr. Wong, I think there's no further questions. I thank you a lot for your presence, for your dedication, and we will contact you shortly. Whoever is interested in collaborating with you, we will communicate that, and we'll see you later or hear from you later. Thank you so much.
Thank you. See you all.
Bye. See you.
Muchas gracias a todos. Los esperamos en las próximas mesas temáticas que sean de su agrado y elección. Y nos vemos después de eso el día de mañana a la misma hora de inicio. Muchas gracias. Hasta luego.